we'll just wait for like 5 minutes and then we'll start with the session, okay? Okay, so we'll start with this week's session, and I'll just give a brief introduction. I am Prashasti Pandey, and I'm a PMRF research scholar working in Department of Botany, Banaras Hindu University. And today is the last and additional discussion session on the coursework, the Swayam coursework, Plant Developmental Biology, whose course instructor is Professor Sri Ram Yadav from IIT Roorkee. And like the previous 4 weeks discussion sessions, in this session as well, we'll be taking few questions from previous year's assignments, which will help you do your assignments so far and also will probably help you with your exam questions as well. So we'll just begin with it, and we'll just follow the same rule. Um, just give me a second, somebody's trying to join. Okay, so we'll be following with the same rule as we were doing in last 4 sessions. I'll be showing a question, and it'll be having 4 options. I'll give you some time. If you know the answer to the questions, you can reply Otherwise, I'll answer the question and we'll just go through with the small concept which will be associated with that question. So we'll start with it. And your first question is, which of the following genes is not responsible in maintaining adaxial polarity? Your options are fabulosa, revoluta, crab's claw, and favulota gene. I'll give you 2 minutes. If you know the answer, please answer it. Okay, so I'll answer the question. So let's first try to understand what all these genes are and then we'll get to the answer. So coming to the first gene, which is fabulosa gene, also the short form of the gene is PHB gene. It is a type of a transcription factor in plants. Now the role of this transcription factor is to regulate the leaves development and it helps in forming of meristematic cells. So basically PHB is also a part of HD ZIP3 family. It's a type of a transcription factor family, and this particular transcription factor basically influences the formation of the abaxial surface and the adaxial surface of the leaf. So as we have discussed in previous sessions that abaxial and adaxial surface of the leaves are the two upper and the lower surfaces of the leaf, where abaxial is the back side of the leaf and adaxial is the upper side of the leaf. Now, PHB gene is responsible for deciding the fate of the cells, whether the cells are going to become the part of abaxial side or they are going to become the part of the adaxial side. And this it does by directly binding to the promoters of the key genes which are involved in this process, or which are basically involved in the vascular developmental procedure. Now, Why is it written that it is done by binding to the promoters? Is because it is a transcription factor. Now transcription factors cannot work alone. They, because they are transcription factor, they will help in transcription of some other gene. So that is why these transcription factors can bind to the promoters of any gene, and it will help in regulation of that gene. So that is how phb will basically help in binding to the promoters of the genes which are responsible for doing this, and it can regulate the fate of the meristematic cells in determining whether they'll be going to the upper or the lower surface of the leaf. So that is the function of the fabulosa gene. Then the other gene is revoluta gene. So revoluta gene and favulota gene. So basically these two genes have an overlapping functions with each other. Okay, so these genes also play a crucial role in development of the apical meristem, as reported in case of Arabidopsis thaliana. Now, what it does is it also plays a significant role in regulating the cell divisions in leaves and stems. Okay, so it will initiate the development in various in lateral positions as well. And also this is a part of hd zip family proteins, which is also a transcription factor, and both these genes are also involved in determining uh, the uh, the fate of the cells, whether it is going to be abaxial or to the adaxial side, or it will be going to the lateral side. So basically it will determine the directions of the leaves from various sides. Okay, so fabulosa, revoluta, as well as favulota. So all these three genes will be responsible in determining the fate or the sites where the meristematic tissues will place itself inside the leaf arrangement. Now the fourth gene was Crab's claw, which is also called as the CRC gene. So in Arabidopsis, it is it basically encodes a YABI transcription factor. Now this YABI transcription factor, it is very critical for the development of carpel and the nectar part. Okay, so also it is also responsible for the flower meristem determinacy. So it will determine the meristem mainly for the development of carpel and nectar. So basically, when the mutations in CRC gene happens, it will result in the alteration of the gynoecium structure, because we are saying that carpel development. Okay, so carpel is a part of gynoecium. So therefore, crab's claw will basically uh, determine the structure of the gynoecium, and basically if the CRC gene is altered or it is mutated, then it will cause the carpel will be a little bit wider than the normal carpel, and it will be shorter. So the name crab's claw comes from the structure of the carpel which is formed due to mutation of the crab's claw, because the carpel will become more short and more wide, and because of that it will get the carpels will not separate easily at the apex part. So it looks like a crab's claw, and therefore the gene determining it is called as a CRC gene, or the crab's claw gene. So basically, okay, so basically, this particular gene will be responsible for the development of the carpel. So coming back to our question, which of the following is not responsible for maintaining a dacial polarity? So the answer will be Crab's claw, because the other three genes are responsible for maintaining the adaxial polarity, or basically for determining the structure of the leaf, whether it will be on the adaxial side or the abaxial side. Okay, I hope this concept is clear, and we'll move on to the next question. So the next question is, which of the following pairs is example of auxin efflux and influx carrier respectively? The options are aux and YUC, pin and aux, IAA and YUC, and pin and IAA. I'll give you some time. Please answer the question if you can. Okay, so we will proceed with the answer of this question. First, let's try to understand what oxygen influx and efflux is. So basically, oxygen influx carriers will move. So if you try to understand the word that we are using here, which is influx and efflux. So influx will be anything which is moving inside, and efflux will be anything which will be moving outside. So because oxygen is a part of plant, anything moving inside or outside will be with respect to the plant cell. So now with this concept, if you try to understand the meaning of oxygen influx and efflux, so oxygen influx will mean that the carriers are moving the oxygen inside the plant cell, and oxygen efflux will mean that the carriers are moving oxygen outside of the plant cells. Okay, so basically these carriers will be important for the uh, growth and development of the plant, and that's the meaning of oxygen influx and efflux. Now, what are the different kinds of carriers of oxygen influx? So there is one which is called as OX1, also called as LAX. So these are the main oxygen influx carriers, and they are a multi-membrane proteins which are similar to basically amino acid permeases, which will help basically help allowing the oxygen to come inside the plant cell. Then there is another which is called as OX1, which regulates in root gravitropism, leaf phyllotaxy, and root hair development. So root gravitropism means that uh, when you keep a plant in an upright position, the root will always develop towards the gravity. So there was one experiment. You can find a lot of papers explaining this concept. Is basically if you even place the plant horizontally, the root will still start growing towards the direction of the gravity. So this phenomena that happens inside the plants, where the root will always grow towards the gravity, is called as gravitopism. So anything that these carriers where the root gravitropism, the leaf phyllotaxy, and the root hair development happens, where oxygen influx is required and the carriers are required, is being done by the OX1 influx carriers. Then the third type of carrier is LAX2. So LAX2 is responsible for regulating various vascular developments in cotyledons, and LAX3 is involved in lateral root development and apical hook formation. So lateral root development happens in a lot of plants due to various reasons. It could be one of the reasons could be stress, that when the plant is facing stress, it will start developing lateral roots to enhance the absorption amount and the oxygen exchange capacity. So these kind of things are regulated by the LAX3 influx carriers. So there are four types of influx oxygen influx carriers: LAX, OX1, LAX2, and LAX3. Okay, and then talking about the oxygen efflux carriers. So there are three types. One is called as spin form, also called as spin. This is the major oxygen efflux carriers. Then the other one is called as PGP or ABCB carrier. This is the member of this family which are also major oxygen efflux carriers. And the third one is ZM pin 1A. This particular carrier will modulate the oxygen relocation in nitrate mediated root formation. Okay, so these are the three kind of oxygen efflux carriers. So if you look at it, the major oxygen influx carriers are OX, and the major oxygen efflux carriers are the PIN carriers. So coming back to our question, which of the following pair is an example of oxygen efflux and influx carriers respectively? So the efflux carrier is PIN and the influx carrier is OX. So the answer will be PIN and OX. So moving ahead, I hope the concept is clear. Moving ahead to our next question. During establishment of leaf polarity, the portion which is towards the meristem is dash, and the portion which is away from the meristem is dash respectively. The options are proximal distal, abaxial adaxial, distal proximal, and adaxial abaxial. I'll give you some time. Okay, so somebody answered third option. Um, okay, third option is not the right option. So let's understand the answer to this. So leaf polarity is called as the so we are okay. So what she answered was it distal and proximal words are basically used in different it is not the polarity actually. So we are talking about that leaf polarity which is towards the meristem and which is either towards the meristem and away from the meristem. So first let's understand what the two polarities of the leaves are. It is called as the adaxial side and the abaxial side. So abaxial side is nothing but the lower surface of the leaf and the adaxial surface will be the upper surface of the leaf. So the lower surface of the leaf will be the one which will have a lot of spongy mesophils and it will also contain a huge amount of stomatas. On the other hand, the adaxial surface of the leaves will contain a lot of trichomes and palisade mesophyll cells. Now, uh, when she said that distal and proximal side, so basically let's look at the figure 2 here. So distal and proximal side will be when the part which is near to the stem will be called as the proximal side and the part which is away from the stem will be called as the distal side. Now the question was in term of which will be towards the meristem while formation. So uh, when we look at the figure 1 here, so if this is the meristematic tissue, this is how the leaf will be formed. If now if we look at here that the cells which are near to the meristematic tissues will be the one which will later form the adaxial surface. Because if you try to imagine now that when this leaf will form here, I'll just try to draw it. Okay, so when leaf will form like this and it will open like this. Okay, because in future the leaf will become like this. So if you imagine this way, then this upper part of the leaf will be the one which is here right now. And this will, so therefore the cells which are near to the apical meristem will become the adaxial surface and the one which is far will become the abaxial surface. Now the reason why the answer cannot be proximal and distal cell is because, I'm sorry, so because if we look in this way, both these cells are near to the meristematic tissue. Okay, when this will grow in shorter part, this will also be very near to the meristematic tissue. That is why the question will not ask me in this way that which one is closer to the meristematic tissue. The answer will be distal and proximal when the question is that which of the following side will be near to the stem or away from the stem. Then your answer will be proximal and distal. Okay, so we look to the question again that during and also the question is about during the establishment of leaf polarity. Okay, so that means it is talking about when the leaf is not yet formed. And in that part, you cannot determine the leaf, you cannot call the distal or the proximal part then and there. Okay, in that case, the polarity will always be about forming the adaxial surface or the abaxial surface. And then also you can understand from the next part that the next part says the portion which is towards the meristem. Okay, if the question will would have been which is towards the stem, then your answer would have been distal and proximal. Okay, but now it is asking which is towards the meristem. So towards the meristem, your answer will be adaxial and abaxial side, which is away and towards the meristem. If it was only stem here, then the answer would have been that towards the meristem will be and also then it will not be distal and proximal. In that case, towards the stem it will be proximal and away from the stem it will be distal. Okay, so in that case the option would have been one, but here in the question your answer is fourth option, which is adaxial and abaxial side. Okay, so I hope this is clear. We'll move ahead with our next question. Which of the following protein is directly responsible for the nuclear degradation during seep element development? The option is NEN, APL, CO, and CUC2. I'll give you some time. Please think and answer. Okay. So let's understand this question. Let's first understand what sieve element is. So there are two main parts of the plants. One is called as xylem and the other one is called as phloem. And sieve elements are parts of basically phloem. So now, talking about the properties of phloem. So basically, phloem helps in transport of food and water, and the movement is in two ways. So it can either go upwards or it can go downwards as well. And then it is so this phloem is composed of sieve element cells and the other one are the companion cells. Okay, so sieve elements basically connect together and they form a tube-like structure. And this connecting sieve cells basically share a highly perforated sieve plate. So if you look here in this figure, so this particular part is the sieve, just the tube. So if you look from here to here, this will form a tube structure, and this is called as the sieve element. And these are the perforated sieve plates. Okay, so because these plates are like the tubes are quite large in diameters, and to support these plates, there are companion cells. So after every sleeve plate, you see there are some or the other companion cells which will basically fit in the places in between. Just give me a minute, somebody's trying to join. Okay, so this will basically help in keeping the companion cells, keeping the sieve elements in its place. So that is the function of the companion cells. Also, companion cells will help with the loading and unloading of the material that is being transported through the sieve element. Now, this movement, basically the sap movement which happens through the sieve element, it is mediated by a hydrostatic pressure from the xylem. So now that is the phloem property, and now that we know that sieve element is a part of phloem. Now the question here was that which of the protein is responsible for nuclear degradation during sieve element development. So that is the question is about when the sieve element is being formed, which gene is responsible for which protein is basically responsible for the nuclear development. The answer to this question is NEN proteins. Now, what is the function of these NEN proteins? So, NEN proteins are identified as the key players in controlling the degradation of the nucleus, which happens within the developing sieve elements. So now this is the crucial step, because here it is exactly at this step where the maturation of the sieve elements happen, and they basically lose their nucleus and then become highly specialized for this nutrient transport. Okay, so uh, then proteins get activated, and that is when they start degrading the nucleus of the cells of the sieve elements. And once the nucleus of the cells in the sieve elements are lost and they are degraded, that is when that particular tissues will become specialized in the nutrient transport. Now, how this exactly happens? What is the mechanism behind the development of the sieve element? So basically, these NEN proteins have the exonuclease property. That is, they have the exonuclease domain. Now, because they possess this exonuclease domains, this will allow them to actively degrade the nuclear material. And once this nuclear material is degraded, it will eventually dismantle the nucleus during this formation of the sieve element. So that is how this NEN protein works. So if you put it in short, the NEN proteins are nothing but the exonuclease kind of proteins which will degrade the genetic or the nuclear material. And once all the nuclear material is degraded, eventually it will dismantle the nucleus. And that is how, once the nucleus is lost, those particular cells whose nucleus is lost will become sieve element and differentiate into forming the sieve element. Now the question is that how is the activity of NAN proteins regulated? Because since it is a protein, it will be definitely regulated by something which will hint it or direct it towards or maybe signal it towards going through this uh, uh, entire procedure of working as an exonuclease and then degrading the nucleus and helping in formation of the sieve element. So the activity of NET protein is regulated by transcription factor, whose example is NAC. So NAC is a kind of transcription factor, and as we talked earlier, any kind of transcription factor will bind to the promoter of the gene, and then it will help in the transcription of that gene. So in this case, the that transcription factor will bind to the promoter region of the NEN gene, the gene which is responsible for producing the NEN proteins. And when this NAC will bind to the promoter, it will help in transcription of the NEN gene, which will eventually produce the NEN proteins. And once the NEN proteins are produced, these proteins will act as endonuclease. It will act as exonucleases. It will degrade the nuclear material, dismantle the nucleus, and then this dismantling or the loss of nucleus will help the cell become specialized for nutrient transport and will eventually form sieve element of the fluid. Okay, so that is the entire mechanism of how the sieve element is formed. Now coming back to our question, which of the following protein is directly responsible for nuclear degradation during sieve element developmental? The sieve element development, the answer will be NEN proteins. Okay, I hope this concept is clear of the sieve element. We'll move ahead to our next question. Members of which gene family typically express in every floral word and gives floral organ its identity? The option is A class, B class, E class, and C class. Okay, so let's understand this question. So to understand the class of the genes which regulate the flower development, we'll have to understand the ABC model of the flower development. So what ABC model tells us that there are basically, let's just call the three genes which are responsible for generating a flower as A, B and C. Now the way these genes are placed here is the C gene, A gene and the B gene is placed here. Okay, so the reason they are placed in this way is that this means that when only C gene is expressed, that will help in the formation of the gynoecium. Gynoecium is also called as the carpel. Okay. Now, when B, C and B gene will express together, then it will form the androecium, which is also called as the stamen or the male part of the plant. Okay. Now, when A and B gene will together express, in that case, it will form the corolla part of the flower, which is also called as the petals of the flower. Okay. And when only A gene will express, that will form the calyx, also called as the sepals of the flower. Now, if you understand from this figure, that in figure A, we are seeing that A, B, C, and there is another gene which is called as E gene. So when all the four genes are expressing, the flower is all in intact position with having androecium, gynoecium, corolla, and sepals, everything. Now in second case, the gene A is not expressing, but B, C and E are all expressing. So when we go back to this figure, when we say that A is part is not expressing, that means the sepals of this flower will be missing and rest everything will be there. Okay. Now in third case, the B gene is missing from here. Okay, here A gene was missing and in the third case B gene is missing. So when B gene will be missing, that means no androecium and no corolla. So only gynoecium and sepals will be there in this case. Now in fourth case, if you see that the C gene is missing here, and when the C gene is missing, then in that case there will be no gynoecium and no androecium, because androecium will only form when B and C express together. So when C is not there, there will be no gynoecium, there will be no androecium. That means in this case the flower will be a sterile flower. Okay. And when none of these genes will be expressing, in that case the leaf will just be a floret of leaves. Okay, it will be a leafy structure and nothing else. There will be no androecium, gynoecium, corolla or sepals. There will be no part of flower in that. It will just be a bunch of leaves. Okay. So now if you see this figure, there is one gene which is getting expressed everywhere, that is E gene. E gene here also, E gene will be here also, and E gene will be here also. And here when E gene is not there, the flower only is not getting formed. So our question was that member of which gene family typically expresses in every floral world and gives floral world organ its identity. The answer will be E class, because E class genes, if they are not present, the flower itself will not be formed and there will be no floral organ. That is why to give floral organ its identity, E class genes have to be expressed, and therefore the answer is E class genes. Now our next question is, cells found around the stomata which regulate the pore size is called as dash: bundle sheath cells, lenticels, palisade cells or guard cells. Okay, so answering this question, the answer is guard cells. So guard cells are nothing but the cells which surround the stomata, and the uh, basically it will help determine whether the stomata is open or the stomata is closed. So each stomata is surrounded by a pair of guard cells which can change their shape and volume in response to environmental signals. So there can be various environmental signals, like it can be touch signals for some case, it can be stress signal, or when the temperature is high or the transpiration rates needs to be increased or it needs to be lowered. In that case, these stomatas will change their volume and shape, and this will determine their opening or closing of the stomatal pore, which will also control the exchange of the gases between the plant and the atmosphere. So the stomata of the plant is responsible for exchanging of the oxygen and the carbon dioxide of the plant. When the stomata is stiff and it is basically in its turgid form, in that case the stomatal pore will be open, and when the guard cells become flaccid or there is loss of water from the guard cells, in that case the stomatal pore will become closed. So the key points about the guard cells is knowing the function of the guard cells, that it can regulate the size of the stomatal pore by changing its turgor pressure. The mechanism is by when the guard cells basically intake water, they become turgid and swell. Now because of that swelling, the pore tends to open, and when these guard cells will lose water, they become flaccid and the pore tends to close. Now, as we mentioned that there are certain environmental signals which can trigger the opening and closing of these stomatal pores. These signals can be light, it can be CO2 level, it can be humidity, and these all factors will help adjust the stomatal opening, whether it will be closed or open. Okay, so I hope all these questions will help you in your exams of this particular coursework. If there is anything that you need to discuss or you want to ask anything, please go ahead. Otherwise, we'll end the session.